Today we will continue discussion about what insights can be gained about the evolution and genomics of the virus by deep sequencing, specifically looking at intra-host variation, and we'll begin a long discussion on the immune response of human hosts to SARS-CoV-2, which will start today, will continue next time, and we'll probably have another lecture after that to specifically discuss vaccine designs and implications, complications, and possible candidates. So today, last time we talked about sequencing and diagnostics, and at that time we had sort of referred to a viral sequence. So the assumption was that there is a single viral sequence that appropriately represents the virus infecting a specific individual. So we discussed that viruses isolated from different hosts will be different, generally speaking, just because they mutate and accumulate mutations differently. Viruses isolated from different species are most definitely different, a lot more so than viruses of the same species, viral species isolated from humans. But if you look a little bit deeper, if you actually zoom in, then you will realize that a viral sequence is somewhat of a misnomer, as viruses do not exist as clonal populations of identical sequences, and they don't exist as a single sequence. They exist exactly as populations, and populations comprise individuals. In this case, individuals are viral particles. These populations are very large, so millions or billions or trillions of viral particles. For example, it is not uncommon I'm not 100% sure what the standard viral load measurements are for SARS-CoV-2, they haven't really been standardized yet, but for viruses where they have been standardized, like HIV, for example, it is not uncommon to have 100,000 copies of virus per milliliter of blood, right? And you have 5 liters of blood, so you can make the calculation that's billions and billions of viral particles. Now, within each of that particle, there's a viral genome. And if you remember when we talked about lecture 2 about what viruses do to replicate themselves, viral replications happens independently in different cells. Within cells, you can make different viral particles. And at every stage, there's an opportunity to introduce error because of fairly error-prone RNA replication and synthesis. Even though there's error correction in SARS-CoV-2, the mutation rates are still quite high. So you can basically assume that viruses will contain some errors which will exhibit as differences. For SARS-CoV-2, if you assume they all start with a single viral particle that establishes the infection, in the beginning there's only one, and then as it starts replicating, you get more and more and more copies, and eventually they'll acquire more and more and more mutations. But it's actually the virus is not going to have that much time within a single host to diverge, because if you think about it, a typical course of infection, as we understand it now, is a couple of weeks. So there's only so many generations of the virus that can occur during that time period. But the viruses exist as a population. However, most viral sequences deposited in databases are consensus sequences, which simply means you take what is the most common nucleotide at every single position. And if you recall, deep sequencing, actually one of the applications when you want to derive a consensus benefits from this majority rule, because that way you correct errors, but you will also remove minority variants by doing so. So you basically represent all viruses with the average most common virus. It's sort of losing unnecessarily resolution of deep sequencing data, because when done right, it can reveal the structure of the viral population. And as it turns out, understanding intra-host variation is essential to study how the virus evolves in response to host selective pressures and so on. There is a nice illustration under the article called viral quasi-species. The quasi-species is a term that was coined in the 1990s to describe the evolution of HIV within a specific individual. That was perhaps the first organism where it became abundantly clear that you have to do multiple sequences you have to isolate multiple virus sequences from a host to understand what the virus is doing. And now it's sort of accepted that any pathogen that reaches large population sizes and has, some duration within the host is going to accumulate changes. So what you see here is a cartoon where, imagine for a second that we start with a virus infecting a pig, which is in the third panel from the left. So there's a pig, and under you have this sort of canonical cartoonish representation of a viral genomic population. So this bar is meant to represent some abstract viral genome. And if you see a symbol on this viral genome, it means it's a variant. And the different colors and shapes are meant to represent different types of variants. So some might be a particular type of nucleotide substitution, say circles. Triangles might be insertions and deletions. Squares might be longer or stop codons or something like this. But the major takeaway from this picture is that there's a large population of viruses that all carry somewhat different mutations, right? Some are more common, some are less common. And then what happens when you transmit to a different individual? So a couple of things will happen. One, and it's actually a point that's not terribly well illustrated in this slide, is that so there's a population of, say, 10 billion viruses that exist in the pig. When something gets transmitted to a new host, it usually will be just one virus randomly picked out of these 10 billion, loosely speaking. So this is also called a very severe evolutionary bottleneck, where you basically randomly pick one individual. It doesn't necessarily have to be random, but this is sort of something you can think about. You might be transmitting multiple viruses, and one of them just happens to be growing out, but it's not going to be all 10 billion. It's going to be some small fraction of what is available. So once that is now in a new host, now there's a different selective pressure. So within each of, say, now human individual, you have ongoing evolution, transmission, accumulation of variants. And you can also see the same thing in experimental evolution, where you do either experimental evolution just to study what a particular bug does as you subject it to viral pressure, or gain or loss of functions experiments, which we may discuss at a later point. But the main point is just keep in mind that whenever we talk about a virus, it's really a population of viruses that might have a large number of distinct variants within a particular infected host. And the number of these variants is going to depend on two factors, or three factors: population size, or population structure more generally, the duration of time, or how many generations the virus is in the host, and the mutation rate, and selective pressures and so on. But there are a lot of variables. So there's a sort of beautiful classical example where people, and again, this is quite old, this is from 2002 in Nature Medicine, and there was a study of SIV, which is simian immunodeficiency virus. And it was sort of an example of where you can use rapidly evolving populations to observe evolution take place in nearly real time. So viruses and some bacteria are often called measurably evolving populations because the time scale during which they accumulate mutations is sufficiently short that you can observe them in experiments. So a classical example that was presented in this paper is immune escape from cytotoxic T lymphocyte responses in HIV. We'll talk about CTL responses later in this lecture, so let's just keep this picture in mind for later on and don't really worry about it too much. And this is a setup where you infect, in this case, macaques with known viruses, and you select these viruses in a way, so you pick which SIV strains go into the macaques, and you pick them in a way that they contain a particular signature. In this case, what's indicated in the yellow box, this epitope, this linear peptide called STPESANL. This is just a collection of, in this case, 8 amino acids. And it is also known that the immune system of these two monkey hosts was able to target. So it recognizes this, and if a cell is infected with a virus that has this signature, it will be targeted for killing. And then they infected it with this viral strain that's at the top. Then they waited 2 weeks, and they sequenced using much, much more laborious techniques. This was before next generation sequencing, so that to use something called cloning and Sanger sequencing. So there's only 9 sequences from one monkey and 10 sequences from the other. But it's remarkable that this is only 2 weeks of evolution, right? So this is really, really fast. And within these 2 weeks, in both monkeys, the viruses that all started looking like this changed. So in the first in the first monkey, 7 out of 9 viruses acquired this mutation in L. And in the second monkey, there was a large variant, sort of different mutations, but every single virus that they isolated had a mutation there. So this is an example of where you have a very strong selective pressure on the virus. It says if you carry this particular combination of amino acids, the immune system can kill you. The virus keeps generating different mutations as it evolves within the host, and the ones that just randomly happen to be in this window happen to have higher fitness, so they grow to higher frequencies. But it's remarkable, just keep in mind, this is 2 weeks, right? So you, in certain situations, you can have very, very, very rapid escape. HIV is very different from SARS-CoV-2. For one thing, it's chronic infection. It has much higher mutation rates and so on. But the point stands nonetheless. So let's actually look at SARS-CoV-2 and try to get sort of a picture in our head of what intra-host variation might look like. And at your leisure, you may want to go and explore a notebook that's linked here. There's something that I wrote just to visualize some of the work we've been doing with collaborators at Penn State and Johns Hopkins to do deep sequencing analysis of SARS-CoV-2. And this is just a simple way to visualize a particular sample, in which case deep sequencing data, from a specific individual. So this number here, SRR 117-28609, is an accession number from the short read archive. So you can type it in and see what this corresponds to. This is a plot that on the x-axis has the genomic position in the SARS-CoV-2 genome. Remember, it's about 30,000 base pair long. So, you start here at ORF1, spike, and various accessory genes. And each of these bars indicate differences from the reference genome. So everything is always, for these types of analysis, when you talk about variants, you have to say variant relative to what? And the relative to what is the reference genome, so the Wuhan strain that is in GenBank under the reference genome. So if you look at what's in this virus, you will see that there are 5 positions, so 1, 2, 3, 4, 5, where 100% of the reads, deep sequencing reads that map to this position, are different from the reference. So these are fixed differences, right? Basically, every single virus that we measured in this individual has the same differences from the reference. So, and you can see what those differences are. So orange means C to T, blue means A to G, and teal means G to C. So there are 5 differences with consensus, away from consensus. So this basically means that the viral population, this individual doesn't seem to have a whole lot of detectable variation. So everything we measured is the same genetic complement, and it's 5 positions different from the reference. If you actually go and look up the succession number, you will see that this was a, let's see, an Arctic Protocol V3 Illumina sequencing data set. It had, 117 million base pair. And it was, because it's, it's an amplicon, so it's only going to be, it's just amplified SARS. And there's, NGS of PCR tiled COVID-19 virus from clinical samples. So amplicon sequencing. You can zoom in and look at it, but, this is one example. Here's a slightly different example, which is a different accession number. And again, it's the same exact visual representation where you have the genome, and bars represent differences from the reference genome. But you will now notice there are 2 different types of differences. So you have 4 bars, this one, this one, this one, and this one, that are basically at 100% frequency, meaning that every single read that mapped to this position has this difference. But there's also a smattering 4 variants that are at intermediate frequency. So not that important, not terribly important what it is, but looks like it's somewhere between 10 and 20%. So for example, this read, roughly speaking, occurs in, this variant occurs in about 10% of the viral population. So one way to think about it, if you take this individual and randomly draw a virus, in 1 cases out of 10, it's going to come up as having this mutation. And there are other variants that are present elsewhere in the genome. So there are 4 differences. So this already tells you that, assuming that the sequencing protocol isn't too noisy, that there are actually different viruses circulating within this individual. For example, you might think that, the original infecting virus had these 4 differences that, one of them arose and it wasn't sufficiently deleterious to be purified. So it's, and you don't know where it's going. You need to actually have time series. It might, if you waited longer, this might grow to fixation, this might disappear, but there's evidence of variation within this host. So we already see that it shows up directly in the data. And it's exactly the same sequencing protocol. So again, Arctic protocol v3, a similar number of reads. It's actually even from the same lab. It doesn't show it here, but it's from the Utah reference lab. So this is, sequencing that they did during, relatively early in the outbreak. And here's an illustration that shows even more minority variants. So now you have 1, 2, 3, 4, 5, what look like major variants. And there's a whole bunch of, probably something like 15 or 20 low frequency variants. And they're actually, there's more that you can go, if you're interested, you can go and explore it. There are papers that have been published on it. And this is a different sequencing protocol, not that it matters that much. So this was sequenced in Washington, so University of Washington, but it didn't use the Arctic protocol. So there was random hexamers. So they basically did shotgun sequencing. So it's a different approach. So some of it could be due to sequencing protocols, but it's, not 100% sure. So even by this, simple examination of a few data sets, and there are several thousand now, and I'll show you, what you can learn from a larger scale analysis a little bit later, we can see that within specific individuals, SARS-CoV-2 does not exist as a homogeneous population of clonal viruses that are all genetically identical, but it exists as a population of slightly different viruses. And the degree of difference appears to be sort of patient dependent. It's, I can't speculate why it is. But we'll come back to this later on. So I thought it might be useful to talk about some terminology. So when you talk about genomic variation, and this, this slide actually was pulled out from a lecture I had on human genomics. It applies equally well to viral genomics. But there's sort of, 3, well, actually 2 major types of changes that I'll explain what I mean. So single nucleotide polymorphism, SNP, or SNV, single nucleotide variant, is exactly as it sounds. You have a change that involves a single nucleotide. So your reference might have an A, and again, all of this variation is always relative to some reference genome. And in this case, the reference genome is what the NCBI says it is, or it has to be specified. But NCBI is usually, or GenBank is the source of authoritative information about what is the reference genome. So in this particular case, it would be an A to G variant. So this is just a change. Then you can have 2 sort of complementary operations, insertion and deletion. And whether or not it's insertion or deletion is, again, relative to what you have to do in order to modify the reference. So in this case, you have extra nucleotides in your read, which means you have to insert them into the reference. And here you have some missing nucleotides in the read or the alternative genome, which means you have to delete them from the reference. So indel, when you see something referred to as an indel, that's what they're talking about, either in insertion or a deletion. You can be, you can be pedantic as saying indel is an operation that requires both insertions and deletions, but typically if you just have an understanding of what is an SNP or SNV and an indel, you're good to go. By and large, when you talk about viruses, you're going to look at SNPs. Although with SARS-CoV-2, there's already numerous reports of some insertions and deletions that appear to be of high frequency. Some of them are quite long, but there are a couple of deletions that are short and a couple of insertions that are short. So all of these happen. But as far as evolutionary process goes, SNPs are much easier to make. All you have to do is just make a misincorporation error when you're synthesizing or copying RNA. Insertion and deletion involves more errors, right? Insertion means you have to stutter and make extra nucleotides, and deletion means you have to skip something. So they're much more rare events. So SNPs are much more common than insertions and deletions. And we can learn I mean, we don't observe any of this. We have to learn it by analyzing sequence data. And just to remind you what we're doing is we have an annotated reference. So again, this is shown as a human chromosome, but it, you can imagine that this is a viral genome. It's annotated, meaning we have, know where the genes are, we know where the functional sites are. So, basically it's extra information we have about site. And then we have an individual viral genome that we're trying to sequence. We don't actually observe this as a single molecule. If you remember, deep sequencing always involves, generating a large number of typically shorter reads that will cover different parts of these genome. Then they have to be assembled together into the genome. And we basically start with this cloud of short reads, and then we have to identify variants. So variants are identified by, basically statistical procedures. They're fairly complicated, and it's an open area of research. So to this day, people are proposing new and better methods or faster methods or more robust methods for calling variants. But intuitively, it is quite simple. So this, here's your reference genome, and these are individual reads. So again, these reads have been mapped to the genome, so you know where they are, and you know that these positions are all in the same location. So here's the reference G, and here it looks like you have 3 A's and 3 G's. Right. So having maybe some intuition from the little exercise we did last time about error distribution, it kind of seems unlikely that you're going to have 3 identical errors, all going to a G or an A. So you might say that this population, at this site, you have 2 different variants, A and a G, and having no other information, you say their frequency is 50-50. Here you have no variation, so this is all a T, right? So this would be a position with a SNP or a minor variant, and this would be a full substitution where you go from a C to a T with no, so it's a fixed difference. And the main intuition, again, how many reads contribute to your call. So here, if you have all the reads agreeing with the reference and 1 read showing as a T, it's more likely to be an error than anything else. So you need multiple reads to confirm whether or not this is a variant, and the same here. So having no other information, you would say this is likely to be sequencing error, so there's no real variation. And if you look at real data, these types of pileups, this is actually a nice illustration which shows you, a typical data set. So you have some reference genome. This is your coverage. So how many reads map to it? You will see sort of a random distribution of reads. So like this read didn't actually cover the position. If there's nothing indicated in a particular column, it means it agrees with the reference. So you have, and this position looks like, a C variant and whatever is in the reference. Right, so the reference sequence is an A, so you would call it as a mixture of A's and C's. And here's an example of something you would not call as a variant because there are only a couple of reads. They're all at the very end, so there might be some errors at the end of the read, so you would say this is not a well-sequenced region, so you would not call this a variant. Obviously, you don't do this by hand. There's software that does it for you. Like I said, it's fairly involved. All the software that you're likely to use will have statistical models. So we'll compute some kind of probability. We'll perform some sort of test to distinguish variation from the noise, and we'll typically report a confidence level. We're not going to go into those details unless somebody wants to follow up with me, and I'll point you in the right direction. But it's just enough to know that this is one of the fundamental techniques in genomics. Variant calling. We'll come back to this probably in a slightly different capacity when we start looking at the human side of the story, probably 2 lectures. So calling SNVs, you need a minimum coverage to distinguish sequencing errors from real variation. As a rule of thumb, 20-fold coverage is typically recommended for SNP calls. And here's a nice chart from Illumina. So it's a manufacturer chart, but there's no reason to believe that it's not accurate. And it gives you a function of cover. How certain you are in calling a variant as a function of coverage. So for example, if you have only 5 reads covering a position, there's no confidence in calling a variant. If you go up to 15 positions, you now have a 92% confidence. And if you go up to 40-fold coverage, you basically have close to 100% confidence. So if there's a variant detected with a 40-fold coverage, you will be able to call it reliably. You can also use data across multiple samples, something called imputation, to get quality calls with lower coverage. But again, this is more technical than we probably need to be. So calling minority variants is a little more complex than just calling differences in human resequencing, because when you sequence a human individual, when you do human genome resequencing, you assume that there's at most 2 positions. There's a heterozygous variant, and that's the most complicated thing that can happen. In a viral population, you can have complex mixtures. So you can have different fractions of bases. So you can have a population that's, say, 90%, 9%, and 1%. So the 1% will be obviously quite difficult to detect. So if there are multiple bases present in different positions, then the problem is more complex. Now, some of the questions you might have at this point is, how common does a variant have to be in order to reliably distinguish it from noise? Another question you might have is, do you have, sorry, let me see, actually, this might be a good question for me to review. Good point for me to review the question. So, sorry, Chad did not indicate to me that there were messages, so I missed them. So let me go back and review some of the questions. Is the speed of mutation normal for a virus? I presume you refer to the HIV example. Yes, this, so sort of a useful rule of thumb is that during a sort of acute infection when the virus is actively replicating, if it's an RNA virus, every single mutation, every single point mutation is generated every time the virus makes a new generation. So it is, if you think about it, a typical mutation rate might be 1 in, say, 10,000 with error correction in SARS-CoV-2. So on average, every viral genome is going to have a couple of errors. So as far as coronavirus goes, it's actually slower than HIV, but it's still going to accumulate mutations. I understand that you stated the reference genome is determined by things like GenBank. However, if insertions, deletions, and other variants develop fairly regularly, how did they determine the original reference genome? That's a great question. At the moment, there's no good solution. One of the things that happens with organisms that are sort of constantly updated, you typically think of this as a human genome, mouse genome, model systems, is that periodically you'll have a revised genome. So you'll have an updated version. If there's some new sequencing data that correct errors or introduce new regions to it. But for example, with HIV, the reference genome is still the first virus that was sequenced in 1983. So it's never been updated. I don't think it'll be updated with SARS-CoV-2. There is actually a really neat computational approach called genome graphs, which allows you to basically build a reference that incorporates the information from everything. So you sort of have a complete record of everything that's been done. So you encode all the genomes, their similarities and their differences. In that sense, you can actually compare everything to the entire body of knowledge, but it's not been widely adopted. So the question was, could there be a variant for some people in the ACE2 receptor gene that confers increased affinity or tighter binding between the spike protein and the ACE2 receptor, resulting in persisting virus? There could be. We'll have a whole discussion on the human genomics. I don't know off the top of my head if this is a study that was done, but there have been a number of studies where various genomic variants in humans were compared to susceptibility or disease severity. So that's definitely a possibility, but we'll review what that is. So one of the things to keep in mind is that ACE2, for example, is there's probably some variation in it, but it's an essential gene. So you don't expect there to be dramatic variation in humans, because if you have a broken ACE2, you probably don't survive. But there's got to be some variation, and this actually might be an interesting question. You can go and research common polymorphism, and there's a database called dbGaP we can look at it, where you can basically type in the name of the gene, and it's going to show you all the documented variants, and it's going to tell you if any of these variants have known clinical or medical significance. All right, I think these are all the questions. I'll keep the chat where I can see it. So going back to this, there's a lot of statistical modeling and due computational diligence that you have to do before you decide that a variant that just appears in sequencing is actually real. And there's another layer complication is that you have, in addition to sort of instrument noise, you also have a multi-step amplification, purification, preparation protocol. And in each one of those steps, you're performing a complex biochemical reaction that is also not error-free. So PCR, for example, can introduce errors. PCR can introduce all kinds of random artifacts into your data. So how do you control, how can you tell that a variant that you measured was actually there when you started sequencing, as opposed to it was not there, but it was put there by PCR error? Because remember, PCR makes billions of copies, and even though it uses very high fidelity polymerases, given enough copies, it's going to make some errors. And if those errors just happen to happen early in the replication cycle, they'll get copied all the way through. And fundamentally, at the end of the day, so if you have, say, I tell you that a particular individual has SARS-CoV-2 infection, whether there's 10 variants at 2% frequency or 20% frequency. Does that mean anything? Does it mean that the virus is, evolving to escape from the immune response? Does it mean that, it's super infection or something else? So how do you biologically interpret this variation? This turns out to be the most difficult question, which is actually not being addressed satisfactorily in many areas of viral genomics. So let me now jump to a paper that was published in Clinical Infectious Diseases. And the goal of this paper was to, it was a fairly early study. I think it was done in April or May. And what they did, they basically wanted to characterize within host variation of SARS-CoV-2. So this was a small study. They sampled 8 patients with SARS-CoV-2. The data are available on Short Read Archive. So here's the accession number. It's in the paper. You can go look at it. They did major transcriptomics, which means that they did not amplify or isolate the virus. They sequenced all the RNA that was there. In other, so one of the things that this enabled, one of the additional experiments that they were able to do because of this is to characterize diversity of pathogens that are not SARS-CoV-2 because they sequence everything. On the downside, they had a fairly small fraction of reads mapped to SARS-CoV-2, so you end up with relatively pedestrian coverage of 278-fold for deep sequencing on SARS-CoV-2. So they also analyzed data from 25 patients with community-acquired pneumonia, CAP, which is not caused by SARS-CoV-2, and 20 healthy controls for comparison. And when you do this major transcriptomics, you basically sequence everything, you do some error correction, and then you bin everything based by doing a database lookup. So you try to match every sequence that you have, every short read, with an organism by doing a database search. And then you bin all the human sequences in one bin and all the SARS-CoV-2 sequences in another, and everything else according to its taxonomic level. When you do this, a fair number of reads will actually end up discarded because you don't know where they go. But because it's such a brute force approach, and you can, you have such an abundance of reads that even if 1% or 1/10 of 1% ends up being SARS-CoV-2, that's enough to do some analysis. So you don't have to be particularly precise. So for example, here you have an interesting comparison that you can do because it's a meta-transcriptomic analysis. So one of the things that they did is basically say, among all the reads that we're able to map to something, what fraction of them map to microbes, right? And this would include microbial reads, would include viruses and bacteria. They stratified it based on the health status of the individuals. So here are the SARS-CoV-2 patients, here's some healthy controls. And the third bar are the individuals that had pneumonia, but that was not from SARS-CoV-2. The Y scale is a log scale. So, this is 10%. Sorry, this is actually very odd labeling. So this is a proportion. So this would mean 100%. This would mean 1/100th, so 1%. And this is 1 in 10,000. Right. So among all the reads that were mapped to something, somewhere in the order of 7 to 10% in SARS-CoV-2 mapped to microbes. So viruses or something else. This was much higher than in healthy control and higher than it was in community-acquired pneumonia. So I haven't seen these charts before. These are called, box and whisker plots. So this line, this meant to represent the distribution of continuous values. So this line is the median value. This box is the 75th percentile and 25th percentile. So this called interquartile range. This shows you, the 95% range of the values. And if there are some points that are outside, these are called outliers. So they're sort of near the end, the distribution. And when you have these types of plots, the other common thing to do is run a statistical test, Matt Whitney or Wilkinson or something like that. It's a standard recipe test that says, are these statistically different? So when you will see that indicated as those brackets. So it means that I'm comparing this with this, and this means, here you can look up what this means. It means p-value very small. So the way to read this is that, based on this experiment and this statistical test, there were significantly more microbial reads in SARS-CoV-2 infected individuals than were in healthy controls. You can compare SARS-CoV-2 individuals with community-acquired pneumonia, and it's also significantly higher, and so on. So, and these are, fairly common plots and biomedical literature. So if you haven't seen them, that's what they are. And the second plot is actually looking at how many of those reads that map to any viruses were SARS-CoV-2. So this obviously is now limited to only individuals that have SARS-CoV-2. And there's actually a fair bit of distribution. So median, if you have SARS-CoV-2, then according to this experiment, a median of 50% of all the reads, that they were able to acquire goes to SARS-CoV-2, which means that the rest of it goes to somewhere else. I mean, the range is quite significant from almost nothing to 100%, but median is 50. This is human rhinovirus, so this common cold. This is influenza, and they also picked up a bunch of random things because they just sequenced basically a convenient sample of people that had some disease. So you have, human adenovirus, human coronavirus, HKU1, measles, respiratory syncytial virus, and so on. Now, I'm glad, Hannah asked you a question in the quiz about, what are the benefits, what are the disadvantages of having research published quickly. And if you look at this figure, you might start asking yourself, what are the benefits of doing a small study? Because first, they only have 8 individuals. And second, because they did sort of this metatranscriptomic approach to sequencing, in many cases, they didn't have a whole lot of data. So this is a coverage plot, which is now the genomic position of SARS-CoV-2. Again, this is a log scale. So like in this individual nCoV1, it was great. So on average, you have between 10,000, and 100,000 reads for every position. So it's incredibly well covered. But then it sort of drops down dramatically. And this dashed line indicates coverage of 50, which they consider to be minimum acceptable for variant calling. It's somewhat arbitrary, and you could argue this might be too low. I'll show you in a couple of slides, why it might actually even be meaningless, if you're looking at variation. But there's some, for example, there's one individual nCoV5 that basically did not have usable data, because if you look at this orange line, significant fractions of the genome simply were not sequenced. And if you think about it, this illustrates to you the degree of experimental noise, because they presumably applied exactly the same protocol with exactly the same reagents, exactly the same, lab tech, exactly the same sequencer to, do the same type of analysis. And they obtained dramatically different results in the quality of outcome. So in fact, they had to, they couldn't use 3 of the 8. So anything that follows below that dashed line was actually not used in sequence analysis. So if you kind of look at this, and if you're, if you're not under the gun to publish quickly, you would say, maybe we should go and sequence them again, just to confirm. That would be, for example, my reaction. Right. Well, the other complication might be that don't have enough sample, but this is not a particularly great quality sequencing data set. And here's another way to look at the variants that they found. So they've looked at, I don't know if it's the greatest plot, but it does contain the information of what they found. So this is just a table. Each column is a particular patient. So nCoV1, nCoV2, nCoV3, and so on. So there's 7 of them. So 1 of them wasn't used. And I believe it was the orange guy that wasn't used. Yeah, nCoV5 wasn't used. Each of the rows is a particular genomic position. Right, so nucleotide 376. Here are the variants that they found. So a G to T change. The quantification, so S or NS is synonymous or non-synonymous. Does it change the amino acid or not? This is the genomic region that was in. If it does change the amino acid, it shows you how it changes the amino acid from E to a D. And finally, the last color is how common is this variant. So blue means 0 or not found. The brighter it is, the more common it is. So for example, these red variants are majority. And then dots means that this particular sample did not have enough reads at that position to make the determination. So for example, nCoV7 is pretty much useless because among all of these positions, they only have data to, call one position. So, just eyeballing this, and here's also these numbers finally at the top is how many minority variants were in each of the genomes. So here's was 1, and here it is. There's 2, these guys, presumably this one isn't called. There's 1 variant. It's kind of hard to say because, it shows more than one, so I don't know if these labels agree or not. And this one has 19 variants. Another interesting thing that they note is that nCoV4 and nCoV7 are individuals that were from the same household. And they both became infected, so the hypothesis is that one of them infected the other. And you would expect, this is kind of an interesting control because you would expect them to share the variants, right? If one infected the other, then they're basically as directly related as possible. And unfortunately, because there's such poor quality in nCoV7, you can't really tell if the variants are the same. So you would expect, say, this variant at 10779 to also be present. It looks present, but it's not, confirmable, and so on. So, the only takeaway from this is that there seems to be, a variable number of frequencies of minority allele variants in clinical samples. So the difference, the question is, what is the difference between the genome location of ORF and ORF? No, there's no, I think the question is there a difference between lowercase ORF and uppercase ORF? There shouldn't be, right? So typically, notation in genomics is kind of a sore, point because it's not consistent and people don't use standard abbreviation. So if you go back to original genetics, the distinction was if you use lowercase italics, it means a gene, so the nucleotides. And if you use uppercase capitals, it means a protein product. In this case, both, I suppose, ORF, if you want to be very pedantic, ORF1A and ORF1B, they're polyproteins. So these are genes that actually make individual proteins, whereas these guys are both genes and proteins. But there's really no difference between ORF lowercase and ORF capital. But I can see that. So the question was, is it dominant or recessive? Because if you go back to genetics, the canonical, capital A, lowercase a for alleles means dominant or recessive. No, unfortunately, it doesn't mean anything like this. I never even noticed this, but now that you pointed it out, it's kind of a bizarre change of scale. I would have preferred it to be consistent. So what are the conclusions of this study? Uh, and a lot of it is speculative, right? And again, just like to point out, this is Clinical Infectious Diseases, which is a high prestige journal, and there's a lot of interest in doing SARS-CoV-2 studies, but this basically has kind of very, very preliminary feel to it. It's a small sample, not great quality, sort of not a whole lot of interpretation. But what they write there, I suppose they don't oversell it too much. But the points that they're trying to make is that inter-host diversity could potentially increase the fitness of the viral population. Further studies are needed to explore how this may influence the immune response toward the virus and whether there is selection acting on different strains in the human body or during transmission. Yeah, I'm sure there is, and we'll find it eventually. Found no evidence for transmission of multiple strains for one possible case of transmission. Again, it would have been a great study if they had high quality sequence data, but they didn't. And then the take-home message is, nevertheless, the observation of high mutation burden in some patients emphasized the possible rapid evolution of this virus, despite that its biological significance is largely unknown. So it's a purely descriptive study, right? Take it as it is. There's really nothing there to indicate that this is biologically meaningful. It's just that it's there. But it is there, and as we study it more and more, we'll probably characterize it better. If knowing what we know from other sort of more extensively studied systems, or systems that we studied longer, like HIV, influenza, some of the other viral diseases. So intra-host variation is how viruses acquire mutations that could alter their population level of fitness. So imagine, for example, that there is a mutation that will make all SARS-CoV-2 viruses, say, more infectious. Where does this mutation come from, and how can it spread through the population? It has to arise in one individual, in one virus within that individual. Then it has to grow within that individual to a substantial frequency. That has to transmit onwards, and then sort of continue transmitting and increasing in fitness. But fundamentally, any change that you're likely observed that has the potential to sort of spread through the entire population has to arise somewhere as an intra-host variant. So that's sort of the earliest detection we can have, where most of the variants, the vast majority of the variants we see intra-host is not going to go anywhere because, it's just random noise and it's neither more nor less fit. It's probably not going to get transmitted. But, if we want to sort of catch the earliest whiff of anything that's happening, we want to look at intra-host variation. And then you could also look, at sort of weird cases that do, have a disproportionate effect on sort of viral evolution. So, for example, if you just happen to capture one individual that has, highly genetically diverse variants, this could indicate multiple infections. So you have two different strains at the same time. It does happen in other diseases. We know it happens in coronaviruses and bats because they recombine. And in order for a combination to happen, you need to have two different viruses infecting the same host. And even more than that, they have to infect the same cell in that same host. And because recombination is a known fact in coronaviruses, you know that multiple infection happens at least in bats. So it could happen in humans. And then if you actually have a transmission of mixed viral populations, so instead of having a single viral variant that seeds a new infection, you have a whole collection of them that are genetically different. It could make a harder immunological challenge down the line, because instead of just trying to target homogeneous viral populations, the new host now has to deal with with diversity. And in fact, there's a more recent paper. It's actually, it hasn't been officially published yet. It's been accepted, so it's an advanced access type of thing. And this is an indication. This is a clinical case of one individual that was infected twice with different strains of SARS-CoV-2. So this is a 33-year-old male from Hong Kong that was originally infected in March of 2020. Went to the hospital, had symptoms, was discharged in April. Then went on with his life. Then in August, he was coming back from a trip to the United Kingdom. Underwent routine screening in Hong Kong, and it was at that point they identified a secondary infection by RT-PCR screening. In the second case, it was a completely asymptomatic infection, so without this airport screening, would have never known. But they went on and sequenced. They confirmed, that he really was infected with a different strain. So there were marker proteins available, even though he didn't have any symptoms. And then they sequenced the virus from March and again from August. And you can see that these two viral strains are very different. So it's not like he was infected with this strain, then it went dormant, and then it just came back 142 days later. No, he was infected with a secondary strain that had a large number of genetic differences, which is how you could identify it. And if you look at this tree, just because these isolates are separate, so they have a whole lot of internal nodes separating them. The most recent common ancestor of these two variants is actually quite old. So, the plausible assumption is he probably got it in the UK. I bet it's a European version of the virus, but yeah, we don't know. So multiple infection, repeated infection does happen, which in this particular case tells you that you don't necessarily get protective immunity after first infection. On the plus side, in the second case, it was completely asymptomatic. So for all intents and purposes, this particular individual did not know that they had a secondary infection. So their immune system did a lot better than the first time around. So, had a memory response. But it is possible to become reinfected. And this is actually, this is very common for respiratory diseases for a variety of reasons. We'll come back to it next lecture as well. But, protective immunity against respiratory diseases tends to be more short-lived than protective immunity against bloodborne pathogens or gastrointestinal parasite pathogens. Based on what you're likely, have experienced in your life, so things like common cold or influenza, you do get infected multiple times. It's not apples to apples because, influenza changes, you're going to get different, the common cold might be an infection with an entirely different virus, but nonetheless, it's not terribly uncommon. So nothing unique here again, against SARS-CoV-2. But it's kind of a cool illustration that the only way you could tell that these are different strains is by sequencing. And doing a comparative analysis. Not only did you need to isolate sequences from this individual twice, but you also need a collection of background sequences to tell you that they're different. To summarize this, if reinfection is common as it is for some respiratory viruses, not clear for nCoV at the moment, then one of the implications is that herd immunity may be unattainable. But on the plus side, the secondary infections could be milder than primary infections, which is, for practical intents and practical purposes, is very useful. So vaccines against some strains may not provide lifelong immunity, which is also common for respiratory viruses. And the one that I think is the most interesting and impactful is that vaccine studies should also include subjects who recovered from COVID-19, to see if they basically, if you're indicated to receive a vaccine, even if you're convalescent after COVID-19, after you had an active infection. So here's just a word of caution. Let's see. All right. I'm going a little slower than I thought. So I'll spend a couple of minutes on this slide. This is, work from HIV folks. As a disclaimer, Ron Swanstrom, the author in this paper, is a long-term collaborator. So we work together still mostly in HIV. But he, his lab spent a lot of time looking at the potential for of deep sequencing to discover minority variants, and especially in the context of drug resistance. And one of the things that he has very clearly shown is that unless you take significant experimental care, you, even if you do even otherwise properly executed experiment, you will find random variants that look quite real. So here's an example, just look at the charts. They did sort of a control experiment where you sequence, where you put what should be a completely identical viral population. So you basically take a virus and you use, stock reagent to basically sequence multiple copies of genetically identical virus. And what you're trying to do then is these guys now, what's shown here is a particular drug resistance variant. So, say K103N means that there's a mutation at position 103, N is, in reverse transcriptase of HIV that is a known drug resistance mutation. And this was done in triplicate, and this is a homogeneous population. So the correct answer is there's no variation whatsoever. So it's all identical. But without any careful experimental controls, you will see that even if you set, say, 1% as is a common cutoff for variance, you will detect variants where aren't there, just because of experimental and PCR noise. And the second one below it is something that's a different experiment where they used a laboratory technique called primer ID, which I'll show you a little screenshot in the next slide, which allows you to get rid of most of the errors. And you can see it gives you the correct answer. There's no variation. Now, if you then go on and try to sequence a clinical sample where you don't know what the truth is, again, you observe, so here you would call, this variant and this variant. But if you do, error correction, you realize that there's only a couple of variants that are there, but another, all the others are errors. And they have this nice, it's actually a fairly difficult concept to explain. So people that do it all the time, in clinical applications, they still, get confused. But, the argument is, so everybody looks at the depth of coverage, but nobody actually looks at how many different viruses you sequenced. So the analogy that they have is, how many pages are in the book? Let me look at the last page. There are 500 pages in this book. Now I'll go make 100 copies of 5 pages and put them in the stack. How many pages are in the stack? There are 500 pages in the stack. How much information is in the stack compared to the book? There are 500 pages in the stack, right? So, and what this means, is that if all you did with your PCR sequencing is, make lots of copies of the same virus, then you don't have as much information as you think, right? So you didn't sequence, say, 10,000 different viruses. You sequence many copies of the same virus. The question is it quality over quantity kind of analogy? It's basically, so there's this process called PCR resampling, which basically means that if you put, and this usually manifests itself if you have very, very low concentrations of virus. So when PCR starts, if you don't have a whole lot of viral copies, what will end up happening is that very early in the process, a couple of viruses are going to amplify early, like 1 or 2 cycles before everything else. And because they have this head start, they're going to appear to be a lot more abundant than they really are. And another way to think about it is that, so imagine sort of 2 extremes. Imagine, you're sequencing, so you're putting something into a PCR reaction, and imagine that you only put 1 virus into this reaction, and say your PCR is good enough to make copies of this virus, right? So PCR will produce, 10 billion copies of the identical virus, but it's just 1 virus, right? So there's nothing you can learn about what's in the population because you only put in 1 virus. So another question, are there any patterns of variation resulting from the experimental PCR noise, or is it completely random each time? There are not a whole lot of detectable patterns. So it's actually very difficult to, do an analysis after the fact to separate error from reality, which is why, if you really want to be diligent, you want to actually put something into your protocol to correct for PCR noise. Did I forget to include the primer ID slide? I think I did. Sorry about that. All right. Anyway, so there is a solution. You can make your experiment more complicated, but you can actually correct for some of these errors. Without getting into too much detail, all I really want you to think about this is that whenever people talk about using deep sequencing data to do sort of very, very high resolution analyses of something that happens within a host, always take it with a grain of salt, because most of these studies will not do all the steps that you could do in order to make your data as clean as possible. Some of it has to do with just, you have to do more complicated experiments. And some of it has to do with the fact that, not everybody is aware of all the appropriate analyses and statistical corrections. But just take it with a grain of salt. And it has to be, the easiest way to decide if something is real or not is just to wait for it to be replicated. Because then you rely on the law of large numbers. With enough noise, it's not going to repeat the same way. So if enough people get the same result, unless they all make the same mistake, it's probably true. So let me give you a little bit of flavor of what else you can learn by doing inter-host variation. So what can we learn about mutational patterns in SARS-CoV-2 by looking at a whole lot of SRA data sets? So this is, there's a link to a post on Virological, which is a viral blog run by the same people that started Arctic. Remember the network that designed all the primers and protocols for sequencing. And again, it's something that we did in with Penn State and Johns Hopkins. So it's a preliminary analysis of SARS-CoV-2 inter-host variation. It includes only SNP variation, so no indels and Illumina data. And it was data publicly available from Short-Read Archive and a European Nucleotide Archive between the beginning of the outbreak and May 17th. So it's about 1,100 deep sequencing data sets. So the first question is, what did the map of inter-host variation look like? And that's what it is, right? So again, you have a genomic position on the x-axis. You have allele frequency from 0 to 100%. And you can see that there's basically, there are some patterns. I'm not going to, you shouldn't overinterpret them, but you can see there are a couple of things you can take away from this. There appear to be sort of clusters of changes. So for example, here's one hotspot, here's another, here's another. So another thing is the color. That basically, so the strategies would mean, so what we try to do is we basically try to look for confirmation. If you had multiple studies reporting the same variant, if these multiple studies use different sequencers, that would be even more information. If these studies use different amplification strategies, so say shotgun sequencing or metatranscriptomics versus amplicon sequencing, the more different studies give you the same result, the more confident you are that these results are real. So this is what the color means. So 1 means only one of the strategies gives you that, and black, so 5 means the most confident one. So they're all there. So 4, and then missense means that you have a non-synonymous change, right? So anyway, so there's variation all over the place. There's question to minimize errors during PCR. Why isn't it plausible to use polymerase with 3 5' exonuclease activity? Well, they already use polymerases that are used in PCR are already very high fidelity, so I don't think you can really improve upon them. And there's actually, the way actually, let me just come back. I'll add a couple of slides on primer ID at the next lecture, just explain it then. But you basically just add a molecular tag to each template. So you add a few more nucleotides to each template. You can then read off, and you can say if these different PCR products have the same tag, then they came from the same template. So you can collapse them all together and correct errors this way. It's actually been discovered multiple times. One in the context of HIV, and it's also very, very common in cancer genomics, where you try to find very, very low frequency variants when you're sequencing, healthy DNA and cancer DNA at the same time, and you're trying to pull out very, very rare variants with high confidence. So the other thing you can say is if you look at all the possible 16 changes, or I guess 12 changes, 1, 2, 3, 4, 5, 6, 7, 8, 9, 11, 12 nucleotide changes, because it doesn't change to itself. Which are the most common? And you can see a striking difference that the most common by far change is a C to T substitution or variant, which is over 100 among all the variants, and C to G is once, as observed once. And then the other is, the most common is T to C. There's actually a pretty good explanation as to why you have such a strong bias. I'll give it to you in a second. But you can already see that there's, it's clearly non-random, right? So you don't, that this degree of difference indicates to you that SARS-CoV-2 does not, generate or tolerate all mutations at the same rate. So C to T and T to C are much more common than, say, CG changes. So the total number of C2T changes is 215, which is 47% of the total, right? So, and the reason you do C2T and G2A is because they're complementary changes in different strands, because, if you're doing standard sequencing, it's going to have, both forward and reverse reads. So, you have to orient them in the right direction. And in addition, there are A to G changes along with complementary T to C, which is 24% of the total. So about 75% of the total are 4 changes out of 12. And one of the interesting things is a lot of these can be tracked to something called RNA editing, which are antiviral host defense mechanisms. And there's a whole lot of them. And, we didn't know about all of them, or probably still haven't characterized them entirely. And this is not specific to SARS-CoV-2 or any particular virus. This is just a general cellular mechanism to, modify or degrade a specific type of molecule. So, for example, there's this protein called ADAR, which recognizes double-stranded RNA, and its job is to basically, look at this double-stranded DNA and hypermutate A to an I, adenosine to inosine, which is read as a G. And it does it randomly. So there's a good example here where you have, this polymerization going on, and there's this ADAR comes along and randomly changes some of the bases. Interestingly enough, it's also being not exposed, but proposed as an RNA editing mechanism, similar to CRISPR-Cas, which I'm sure some of you have heard of. It's just a way to engineer specific mutations into the genome. And ADAR editing is also prevalent in human expression data. So there's actually, if you're curious, there's this paper that goes through and finds there's a lot of T to C and A to G hypermutation in especially human non-coding regions. And there's another one, this whole family of enzymes that was, characterized fairly extensively about 20 years ago, because there was a lot of excitement. This could be one avenue to cure HIV/AIDS. ApoBecs, so these are deaminase enzymes. So what they do is they also do, but they do single-stranded DNA editing, and they do C2U deamination. So A to G or C to T hypermutation. This appears to be a very ancient antiviral defense. So it goes back, all the primates have it, past the primates, I think, all the way up to about 60 million years ago. These ApoBecs are present in different species, and in fact, they seem to have evolved together with viral antagonists. So this is a really ancient mechanism. But if you think about what this would do, this basically, it's not specific against SARS-CoV-2 or anything in particular. It just generically sort of messes up RNA or DNA. And not all of it is even necessarily antiviral activity, but it's just a way to, edit, perhaps degrade RNA. But so how can we tell if variation is due to this RNA editing as opposed to mutation? And there's been a lot of research where people have done experimental or computational analyses to realize that, in order for these enzymes to be somewhat specific, they can't just randomly attach to RNA molecules. They typically recognize specific motifs. So a particular sequence of letters within an RNA or a DNA molecule is where they bind, and then they make a change. So for example, for ADAR, it seems to matter what is one nucleotide after and one nucleotide before what you're trying to edit. So here's, sorry, these are very ugly charts, but they're not mine. I just pulled them from the paper. It's referenced, all of this is referenced in the virological post, so you can go and dig out the original references if you want. But basically, there's if there's a G following the edit base, the ADAR enzyme is much more likely to make an edit. And it also doesn't like G's before that. It prefers, C's or a T's. So with this in mind, we can basically say among all the changes that could have been ADAR editing, so all the A to G changes, what is one base before and one base after? All right, so the most common is a G to A, which I suppose you could argue is a motif, but it's kind of I think a stronger motif is ApoBec. So ApoBec likes motifs like TC, CCC, and TCC, where the last base is mutated to T, or TCW. W is basically a shorthand for an A or a T, but the middle base mutates to T. So if you stratify all the changes from C to T, which if you remember are one of the most common changes in the genome, the second most common is an apobec signature motif in one strand, and the two most common in the other strand are also apobec motifs. So we don't have any direct evidence in this analysis to say that it's RNA editing, but RNA editing definitely seems like it's a significant contributor to genomic variation. So we published this in May, and a couple of months later, or we posted it in May, we didn't publish it. But there was a paper, there are actually several papers that specifically test for host-dependent RNA editing in the transcriptome of SARS-CoV-2. So, they mention ADARs and apobecs. And this could be interpreted as one, the antiviral defense mechanisms are actually doing what they're supposed to be doing. And they explain, some of the observed intra-host variation. In fact, you might imagine if they're doing their job, they're going to kill the virus in a particular individual, or make it so mutated that a substantial fraction of the viral particles don't survive. All right, so let's take a recording. So continuing on to what's probably going to take us a little while to cover. I anticipate the remainder of this lecture for sure, and then probably all of next lecture, is the various aspects of immune response to viral infection. And the one thing you should know about the immune system is that nobody really understands the immune system. A good immunologist is likely to tell you exactly that. And they tend to, most immunologists know one part of the immune system quite well, just like any other specialist, but it's both highly complex and highly effective. We're all testaments to the fact that it is effective, because every day, I'm sure a number of pathogens would like to kill us. And there's still many things we don't understand about it. So I'm not an immunologist. I have, just enough understanding of the immune system not to get into too much trouble. And I've, worked with various aspects of immunity, from the standpoint of viral analysis and some vaccine design and immunogen design, mostly in the context of other viruses. A little bit in the context of SARS-CoV-2, but immune responses, it kind of, the natural analogy is the military engagement, where, virus is the enemy and the host is the defending army. They have to go through a series of stages throughout the infection. At each stage, the host puts up a series of progressively more sophisticated defenses. The enemy, or the virus, doesn't come unarmed. A lot of them come with a number of specialized defensive weapons and sabotage systems to overcome the defenses. Some of them we already discussed when we talked about the molecular virology of SARS-CoV-2, kind of looking forward, specific viral factors that suppress various components of the immune response. In most cases, the virus loses. So, it replicates for a couple of weeks, and then eventually gets completely removed by the immune system. Reaches an impasse, which we discussed, which basically means sort of some kind of a dormancy, or low-level persistence, or perhaps flare-ups. It really depends on the virus. In any case, SARS-CoV-2 does not appear to be a chronic disease. I mean, there's some discussion of long-term effects, but they're not viral, not due to consistent continued viral persistence. So in rare cases, the virus will become chronic and can lead to serious diseases or death. And viruses are ancient, so are the defense mechanisms. So presumably, nobody knows how old viruses are, but they're viruses that infect bacteria. They're basically viruses that infect everything that's not a virus. So it's a very ancient branch of on the tree of life, even though one can debate if virus is alive or not alive. So this great overview of viral responses, immune responses to viral infections is linked here. You just borrowed a couple of tables from it. So these are the stages of viral infection. They tend to be shared by all the viruses. And what you'll see in this table is a particular viral event. The human or host defense mechanisms that try to prevent this event from successfully completing, and at what time during the infection does it take place. So the first thing the virus has to do is to actually transmit to the new host. What that means depends on the virus. If it's a respiratory virus, it has to be inhaled and deposited on the right type of tissue. If it's a blood-borne pathogen, it has to be some sort of a fluid transfer. You could, if it's a gastrointestinal virus, you eat it. There's a variety of ways that, sexually transmitted diseases and so on. Many events do not occur because of mechanical and chemical barriers. So the virus simply fails to reach a beachhead, in military terms. So, the invasion fails before it starts. Mucosal membranes are particularly good at stopping these infections. Just to give you, flavor of how, this is a very effective barrier, even though nobody really thinks about it. For example, HIV, which is a famous sexually transmitted disease, is terribly inefficient at transmitting. Presumably, a lot of it has to do with mechanical barriers. So as a rule of thumb, anywhere between, if you have, so the standard route of transmission for HIV is an unprotected sexual intercourse, and you typically need to have between 100 and 1,000 of those to get an infection. So it really, really is inefficient. And, it's not because the virus isn't trying, but it's also because you have, defense mechanisms that are there to stop it. Assuming it is able to establish the initial infection, it now has to, enter the host. It actually now has to start infection and replication. So if you remember what that was from molecular virology, it means it needs to find the right target cells. It needs to bind to the right receptors. It needs to infect the cells and start its life cycle. So, what stands in the way of this is innate immunity, which is a very broad set of molecular mechanisms that are not specific to a particular virus, but they're sort of general, pathogen defense mechanisms which, activate, again, generic responses. And innate immunity is called that because it's an ancient branch of the immune system that's shared with sort of simpler organisms like plants and insects, whereas adaptive immunity came on later. And this also takes place almost right away and continues to be important. So the next step is, does infection, is it stopped by innate immunity? And if it not, is it spread? So the obstacles is then, if innate immunity isn't fully successful at suppressing the virus, now you need to recruit adaptive immunity. And the first thing you need to do is you need to transport pieces of the virus to specialized lymphoid tissues where a lot of the immunological lymphocyte development takes place. So that's what that means. And it happens very quickly. So this is within the first 24 hours. Once this happens, your adaptive immune response is aware that there's a new pathogen in town, and it's going to mobilize its resources and see what it can do. So the next is, supposing this continues to proceed, so the next step is, infection controlled, meaning that the virus, is able to replicate, and that it takes some time for the adaptive immunity to marshal its responses and deliver the right cells to where the infected cells are and clear them. And this is mediated through specific antibodies and cell-mediated immunity, and typically takes 4 to 10 days. And then following this, you basically have two outcomes. So, if you're successfully able to clear the virus, then whatever response was effective is going to sort of become immune memory. And this will, ideally result in full sterile immunity. So that means that you have, next time the same or similar pathogen tries to invade, it's stopped much faster because you already went through the stages of developing a specific response. And depending on how long, the type of pathogen, it could fade very quickly, or it could basically be lifelong. And the alternative is you can have viral persistence if infection is not controlled. This is the least desirable outcome because now you basically have an ongoing battle that can last for a long time. And this basically continues to drain the resources from your immune system. And if it's sufficiently destructive, you can reach a state called immune exhaustion, or other undesirable endpoints, where you could either have sort of continued symptoms, or you can again progress to severe illness or death. Again, HIV, for example, does not kill the host directly. It exhausts the immune system to the point where something else gets in that normally would be easily cleared and kills the host. Question is, could you contract two viruses at the same time? Would the immune response go simultaneously or separately? Oh, you can definitely deal with multiple pathogens at the same time. In fact, it's probably a common state. At any particular time, your immune system is doing multiple battles in multiple fronts. I mean, you get exposed to pathogens, all kinds of things every day. Just most of them don't get to the point where you realize that you have them. And the reason, and you can actually measure this. You can measure co-infection. You can measure what your immune system is responding to, and it's pretty clear that you can have very complex dynamics. What triggers the activation, deactivation of viruses like herpes that are latent in the system? Again, it really depends on the particular viral system. I mean, sort of the standard, for herpes, it would be stress, right? So you have particular cascades that are activated. But typically, if you have a latent virus, it's it's some sort of signal. It really depends on the virus. It's once you get into these adaptations, it sort of becomes system specific. So, phase 0 in this battle, why does it stop replicating and go back to the inactive phase? Again, it depends on the viral system. All right, I mean, one of it is basically you have, so one possibility is that you get you continued immune control, but depending on the viruses, depending on the virus, it basically can sort of be in a latent stage where the immune system doesn't see it. And again, HIV is again a classic example of this, because the reason you can't cure it at the moment is you can treat it, but you can't cure it. And the reason for it is because it's a retrovirus, so it integrates itself into immune cells. And these immune cells are designed, memory cells specifically, to basically go to sleep for many years, because it's their entire job. Their job is to, remember what they saw and then go to sleep, and be periodically activated if something similar is seen. So every time they're activated, the virus gets replicated along with the cellular proteins. But it's very specific to say retroviruses. And one of the things about viruses is that you have they're highly diverse, and we probably know very little about the viral diversity. We've only studied the viruses that infect us or infect systems that are important to us. So cattle, say crops. We're now very interested, for example, in the virus that attacks grapevines, because it causes losses to the wine industry, but we don't really care about the grapes per se. Silly question, but can there be communication between different pathogens of protein production or toxin releasing to aid in cell invasion? Oh yeah, there are definitely sort of synergies and enhancements between different viruses. So one of the things, and again, sorry to keep falling back on the HIV example, but it's the system that I know best. Cytomegalovirus is sort of a common example of something that can enhance HIV replication, because it stimulates cell cloning. So CMV or cytomegalovirus will, for its own reasons, signal T cells and other immune cells to start making clones of themselves. And if those cells happen to be infected by HIV piggybacks on this expansion and gets a lot more copies than it normally would, because it doesn't have the ability to trigger this particular pathway. Is there ever a point at which different pathogens interact and one almost turns off another, leaving only one to infect? I'm sure there is. It's just, I don't have a good example for you. When there's got to be one. There's pretty much anything you can imagine happens. And if it doesn't, it just means we haven't discovered it yet. Because again, viruses are ancient, and we've been co-evolving with viruses for billions of years. So it's kind of a very, a billion years long dance where we kind of all know each other's moves to a point. Phase 0 is physical and chemical barriers, so repel viral infection before it takes hold. So there's, again, a cartoon from this nice review, which sort of shows you the common routes of entry. Again, this is not an exhaustive list, but a lot of the viruses, enter through the respiratory tract. So influenza, RSV, rhinoviruses, coronaviruses, adenoviruses, measles. You have some viruses that enter through the eye, some viruses that enter through the gastrointestinal tract, skin entry and infection, genitourinary tract. You can have more exotic routes of transmission, like eating animals, or blood transfusions, hospital transmissions, but these are the common ones. And there are numerous obstacles for entry and infection by most human viruses. They're mechanical barriers provided by the skin and mucosal surfaces, which are there to basically trap particles before they get anywhere. And you also have chemically hostile environment of the gut. And you also have in mucosal membranes, you actually have specific, very, very active immune surveillance, because that's where a lot of the pathogens are first seen. And again, a lot of the viruses actually encode specific proteins or cascades of proteins that interfere with immune constitution of, say, mucosal membranes. So innate immunity is just phase 1, and I'm not going to talk about it much today. I just want to throw it up there to, this is the simple part of the immune system, the general defensive strategy. But even at this is very complex. So here's a high-level diagram of the pieces that are in the innate immune system. And obviously, I don't expect you to read this. I just wanted to throw it up there to show you that it's a complex system, even at a high level. Innate immunity. So it basically, all immune responses, they're carefully choreographed stages where you have that involves hierarchies of immune cells and signaling to marshal the defenses. One of the things about the immune system, in addition to everything else, is that you want it to be active, but you do not want it to be too active. Because if the immune system is activated when it doesn't need to be, it can lead to poor outcomes for the host. You can have autoimmune disease, for example, or just immune dysregulation. So immune system exists in what's called homeostasis. So if nothing disturbs it, it's in equilibrium, right? Things sort of turn over at a constant rate. You don't have uncontrollable growth of cells or decay of cells, which means you have very sophisticated signaling mechanisms which turn it on when it needs to be turned on. It goes through the process, does what it needs to do, and then it's turned off. So a lot of that has to do with signaling, cytokines and communications and transport. Innate immunity is ancient in the sense that it's shared with more primitive organisms like insects and plants. And very broadly speaking, what's responsible for, it's just responsible for general activation of the immune system, just, signal. It's basically something is here. Let's do, there's an invader. Let's do something about it. One of the hallmarks of it is inflammation, which is a complex response, which is an indication of immune response, and it serves a number of functions like creating obstacles to viral transport, providing signaling for immune cells, replication, whatnot. Individual cells can basically enter special stages, where cascades of genes are activated by the immune response to do things like suppress protein synthesis. So if a cell, if this is activated and it's making viral proteins, then one of the general defenses is they'll just turn off protein synthesis, in which case nothing gets made. The cell can potentially die, but you can live with that as an organism because you get 60 trillion cells as a human, so it's not a big deal if you lose one, or maybe it's 6 trillion. Can't remember. I think it's 6 trillion, but nonetheless, a large number. But the viruses, because of that, will also have viral factors which counteract this, for instance. And then you have these NK cells, where NK stands for natural killer, and it's something that we only I want to say characterized about 20 to 30 years ago. And it's a fairly recent discovery. So, and these are cool sort of general killer cells. And it's also a general defensive mechanism that responds to sort of a specific strategy that a lot of the pathogens adopt. So what NK cells do, they basically surveil for general deviations from normality. And what this means is that viruses or cancers, for example, can alter the normal states of the cell they infect. And one common defensive mechanism of the viruses is to stop the cell from displaying friend or foe markers through these MHC1 or major histone compatibility complex 1 on the cell surface. So in a way, by doing this, the virus says, I'm just going to hide evidence that I'm making viral proteins inside the cell. But because it basically turns off the entire MHC or downregulates the MHC cascade, that will indicate to the NK cells that the cell that's infected is abnormal, not because it's displaying viral proteins, but because it's not displaying enough normal proteins. So it knows that the cell is abnormal. It doesn't know or doesn't care why. And NK cells can recognize this paucity of MHC1 on the cell surface and then lyse or kill these cells. And it's also meant to be a very rapid response cell, so it can and it can basically take care of the infection before it really spreads. So the question is it true that the level of severity of COVID symptoms depends on the type of virus strain a person is infected with? For example, if one person has severe symptoms, it will spread stronger viruses, and other people infected by that will also have more severe symptoms. It's a great question. At the moment, there's absolutely no evidence that the viral strains that circulate are dramatically different in terms of their pathogenicity. There was one mutation. So there are two reasons for it. One, SARS-CoV-2 is actually not that variable. On average, two strains that circulate today will maybe have 10 mutations, which from the standpoint of viral diversity is nothing. It's basically the same virus. It's not enough, right? If you're looking at, say, seasonal influenza, it's a significance like 10 orders of magnitude, possibly more level of turnover. So there was one mutation that people thought might be important because it arose in the spike protein, and it kind of went from low frequency to high frequency pretty quickly. And it had some indications that it might be more lead to more efficient viral replication. But there was a large study done in the UK where they specifically looked to see if this mutation is associated with any kind of negative outcome on health, and there was absolutely nothing they could find. And they had a very large sample size, so it's at the moment we don't have any evidence of that. So most of the variation that you see is probably due to other factors, not necessarily the genetic composition of the virus, even though it probably plays an effect. It's just not a dramatic effect at the moment. Now, as the virus continues to replicate in the human population, traditionally speaking, you expect it to to change over time, because we start putting selective pressures on it. It's going to start evolving in some directions. Fortunately, what has happened in the past is that viruses that get into the human population, initially they actually become less pathogenic on average, because it sort of optimizes their spread. You don't want to kill the hosts too quickly. The viruses have a different adaptation strategy. Their goal is not necessarily to kill the host. Their goal, like everything else that's alive, is just to make more of the virus, to continue spreading. And a lot of these strategies have to do with sort of extending the time during which individuals can be infected or continually reinfecting the individuals. But we'll discuss, we'll spend at least one lecture talking specifically about what is known about the genetic markers for disease severity. I'll give you a little flavor of it in a couple of minutes at the end of this lecture, but we'll discuss that. There's 23andMe just recently announced that they did, I haven't read that study, but I'll tell you what it is once we get to that point. They did some large scale analysis of what correlates with COVID severity. All right, so today my goal is just to give you one study about one branch of the immune system, and specifically the adaptive immunity, which are the big guns. There are two major types of adaptive immunity that raise responses to specific antigens. So everything up to this point is generic. It recognizes specific common markers or specific types of molecules, just foreign something. Adaptive immunity looks for very specific signatures that are unique and characteristic of a specific pathogen. So this will be say a linear peptide for CTL response or a structural motif for antibodies. And they're also responsible for immune memory. And you have two main branches. You have T cell immunity and you have B cell immunity. They're both subsets of white blood cells, so they're lymphocyte subsets. So T cell, specifically, they're the branch called cytotoxic T cell lymphocytes. So their goal is to recognize and kill infected cells. So they don't target viral particles. They target cells that have active viral replication, or something, not necessarily viral bacterial, but something that makes foreign peptides. And the T cell immunity is generally responsible for clearing or resolving infections. So that's what comes up and takes care of clearing the virus if it establishes a successful infection. And then B cell immunity, which is sort of acts through antibodies, and more on this next time. They do something different. They recognize and neutralize viral particles. So the T cell immunity is deal with the infection when it's here. B cell immunity is try to stop infection from happening. So it's also generally responsible for protective immunity because it stops infection. And vaccines will, again, generally target the elicitation of antibodies. So they look for B cell immunity. And antibodies are specific types of molecules that bind to viral proteins and neutralize them in some sense. So they can either stop infection completely. So they basically say they clog up the viral receptor so it can't bind to the cell. That's one example. They can they can attach to the viral particle and then attach to each other to basically make clumps of viruses, so they can no longer infect because they're now in a cluster. This is called agglutination. Or they can tag viral particles for destruction through specific mechanisms like macrophages that, their entire job is to basically be trash collectors, find something that antibody tagged, eat it, digest it, and get rid of it. And T cell and B cells both recognize and target epitopes. So epitope is a term that refers to unique targetable feature of the virus. I'll give you specific examples in the next two slides. So structural sequence pattern. Question is, are there interferons that target the SARS-CoV-2 virus? Are they ever useful to examine for therapeutic approaches? Interferons don't necessarily target a particular virus. They're sort of more of a generic response. It is definitely true that SARS-CoV-2 appears to have factors that repress interferons. So it is useful to examine them in that sense. But, interferons, they're sort of too broad. If you want to look for a specific, to resolve a specific antigen, specific infection with a specific pathogen, you probably want to recruit adaptive immunity that will target that specific response. Because basically, you can think of interferons as carpet bombing, right? Not precision strikes, it just, destroy everything indiscriminately. So it's not terribly useful in that sense. It's, it's not as powerful as a targeted strike, if you like my lame military analogies. So T cell immunity, again, just remember the whole goal of it is to recognize and kill an infected cell. So a couple of questions. It's how do you recognize that a cell is infected? I mean, a cell is enclosed in a cellular membrane. There's something going on inside, but this other cell is outside, cannot peek inside the infected cell. So it needs to know whether or not everything is all right, and if it's not, what is happening. So fortunately, there is a very robust mechanism whose entire job is to basically, inside this cell periodically chop up all the proteins that are being made into short pieces, transport them to the cell surface, stick them onto these MHC molecules, whose entire job is to basically present short pieces of molecules that are being made inside the cell. And if you think about it, most of these molecules are going to be perfectly normal. So whatever the cell is making, in the process of its normal life cycle, but occasionally you'll have foreign peptides. So these cytotoxic T cells have complementary receptors called T cell receptors. Not very creative, but sure enough. And their goal, what they do, they're going to be able to bind to MHC receptors only if this MHC receptor has a particular foreign peptide. So on their own they can't bind. You need to have this piece that says there's something in the cell that shouldn't be there. Assuming that this specific molecule is recognized, then binding occurs, and CTL releases cytotoxic factors, which are shown here as these, beautiful, I guess, particles that are traveling to the cell and going to destroy it. It releases cytotoxic factors which kill the infected cell. Let's see. What portions of the SARS-CoV-2 virus do antibodies target? Can be the spike protein, right? Since it was the case of our body may have autoimmune antibodies that might not be able to differentiate itself from else. Well, no, actually, most of the vaccines that are being trialed now, they raise antibodies specifically against the spike protein. And the reason for it is antibodies bind to what's in the surface of the virion. Remember, they want to neutralize viral particles before they do anything. So they have to be able to recognize what's on the surface. So the most common thing that's on the surface is the spike protein. So that's exposed and it's available for binding. It is not necessarily true that that's the only thing you could target. But for example, if you're trying to target some polymerases with a B cell response, it's not going to do anything because those polymerases are not visible to the antibodies until the virus is in the cell, unpackaged, and is replicating. And then the question is, so as far as antibodies, so the question is, can you raise antibodies that will accidentally target self? You could in principle, but it is very rare, and there is a very, very complicated and strong mechanism that is specifically there to prevent it from happening. So one of the stages in the developing of B cells is a calling stage where if they recognize self, they will be killed before they're released to where they can do damage. If you have some sort of an autoimmune disease or dysregulation of the cycle, yeah, sure, you can have it. And occasionally you do get autoimmunity, but it's not the rule, it's the exception. And is it possible for the T cells to disregard an infected cell? Yeah, so recognition is not perfect, right? But it is pretty effective. And I'll show you in a second, how this, could be problematic for the virus. So again, there's another cartoon of what the CTL cell does. So you have foreign viral peptides. If they're inside the cell, they're going to be cleaved by the proteasome right here into little bits. Those little bits, float around, get loaded into a special transport molecule called TAP, loaded into the MHC class molecule, transport to the cell surface, and then they stack out for surveillance. And if a CD8 positive, a specific type of cell marker that is characteristic of cytotoxic T cell lymphocytes, if it binds to presented foreign peptide, a T cell receptor will initiate infected cell apoptosis. And peptides, one of the sort of easy things about T cell immunity is that what they target is literally just a contiguous piece of the viral protein. So they present linear foreign peptides. So, imagine here's some protein sequence. You just read it left to right, and you take fragments of a specific length. So for example, for CD8 epitopes, it's usually 9 or 10. It's longer for CD4, 15 to 20. But anything, that's the right length could potentially be recognized. So you can imagine, you take a viral protein, chop it up into substrings of 9 letters long. Each one of those could potentially be presented to a T cell, and if it's recognized by the T cell, that'll kill the virus. Obviously, you want these peptides to be sufficiently unique, so you don't want these to be, common in self-peptides, because then you'll have autoimmunity. And this is undesirable. So not everything is going to be targeted. You can also have some degree of tolerance to errors. So, for example, it doesn't have to recognize this specific peptide, but it can recognize everything that's, say, one amino acid away from it. It's going to be not as good, but it'll still be some binding. And say for CD8 epitopes, some specific sites like 2 or 9 are usually more important for binding. So for example, here a mutation of the tryptophan might be a complete escape mutation because it's in an anchor site, but a mutation at another position might be tolerated. And if a virus, if now you can go back to the first example from SIV that I showed you, where you had this type of peptide or epitope that was being targeted, and the virus very quickly developed escape mutation in that epitope because it was a very, very strong selective pressure for it to do so. The question is, so T cells have many TCRs that are unique to each specific sequence that will elicit the same response. No, a TCR receptor has unique specificity. So there's a lot of variability. Different T cells will have different T cell receptors, so we'll recognize different peptides. You don't want to be able to, you don't have a generalist T cell, just like antibodies have generally, they bind a specific antigen. So you have lots and lots of different T cells that recognize specific peptides. In fact, there's a lot of, because they're parts of the same immune cell hierarchy, there's actually a lot of commonalities between B cell receptors and T cell receptors, but they're different between T cells. So as an example, a particular epitope, say a piece of viral protein that are recognized by the CTO response, will be recognized regardless of where it originates. So if another virus happens to present an epitope that the immune system saw in the past, say from another coronavirus or something similar to it, the immune system will recognize it. We also do not want epitopes that come from normal peptides to be recognized, because this would trigger autoimmunity. Mutations within recognized epitopes allow the virus to escape immune response. Epitopes can be predicted computationally and measured experimentally. And important, different people will recognize different epitopes. So as an interesting aside, the whole sort of the first place where MHC and HLA came into medical literature was in the 1960s, when there was active development of organ transplantation. And you will see, if you ever, God forbid, need an organ transplant, you will require a matched donor, a donor that has the same HLA haplotype, which means that the proteins that are in that organ will be recognized as self by your immune system. And that there's, unfortunately, because of this, I think HLA terminology ended up being very, very unwieldy. HLA, is pretty big on immunotherapy question. So there was also a question, what does the orphan 8-mer refer to at the bottom of the last slide? Let me check. Sorry, just ignore that. This was an illustration from a company that makes overlapping peptides. All right, so here's an example of a SARS-CoV-2 epitope, just to give you a very specific example. So there's one epitope, and that's all it is just a fragment of the SARS-CoV-2 protein sequence, AVF, Q, SA, SKI. So this particular position actually has a mutation that changes the Q, so it could potentially, in some sequences, there's, say, 60, in the database, as of a couple of weeks ago, there were 66,500 sequences that present this peptide, and it's targeted by a specific type of HLA allele. So this, designates which host is able to recognize this type of peptide. And this HLA B5215 is just, what is one of the loci, immune loci in our human genome is. There's, HLA-A, HLA-B, C, DR, and so on. There's a bunch of canonical loci. And, if you ever get sequenced for an HLA type, you're going to have a complement of these alleles. And, I have no idea. We have enough people, somebody might have this particular allele, but perhaps not. But one of the key points is that these alleles will have different geographic distributions. So there's a website called Allele Frequencies. So if you're interested to see where how common a particular allele is in different parts of the population, it'll show on a map all the studies that have been done. Blue means it's very, very rare. 1 means it's very common. So this particular random allele that I picked, seems to be completely absent in Europe. In certain population in India, it seems to be quite common. And there's a smattering of it, in New York for some reason, and maybe in Central America, and it's in Japan. So one of the things that might indicate, and again, I'm not saying this is what happened, but that this would be a fairly common thing that viruses do, is that if a virus, say, if this allele is able to recognize SARS-CoV-2 and effectively kill it, and it's only seen in Japan, say, or in India, then the viruses that are in Japan and India will have an evolutionary advantage to produce a mutation in this epitope. Viruses in Europe, there's nothing to them. Fixing random mutations is probably deleterious if there's no pressure to it. So if viruses are all geographically isolated and they circulate in populations long enough for this to work through, what you'll see is that viruses in different parts of the world will end up sort of becoming modulated by the immune response in that population. It's unlikely to happen for SARS-CoV-2 because it's, a respiratory virus that has, very high rate of transmission. And if anything, it travels around the world. But, sort of we'll wait long enough, we might be able to see these changes. So these mutations are, the question is germline or somatic. So these mutations are in the virus. So in the virus, I guess everything by definition is germline because it doesn't have a multicellular structure. These are also germline mutations, right? So, what determines, you inherit your HLA type from your parents. You don't. I mean, in some individuals, you get, very rare de novo mutations in them. So this is, the colon 15 is a minor variant, but it's all germline. So there's, there are a number of studies that have looked at the viral genome and basically tried to use, well-developed computational models that, split the viral genome into individual pieces and then try to predict whether or not the human immune response will recognize them. So there's one paper that did this. Well, actually, that's, what I used in the previous slide to identify an epitope. So they found, in at least 90% population of 57 countries. So what that particular study says is there are a lot of potential targets in the SARS-CoV-2 genome that should be recognized by the T cell response. And this brings me to, the final topic of this lecture. And I'm sure you've heard about this. And if you haven't been paying attention, is there have been a number of papers that got covered in the popular press as there's evidence of pre-existing immunity to SARS-CoV-2. So what I'm going to do, I'm going to give you, a brief overview of one of these papers to try to give you a sense of what they're actually telling you and how to interpret this. Let me take a question. This may have been discussed previously, but do different mutations of SARS-CoV develop in different areas of the world? For example, do mutations in the US look significantly different? Mutations in Africa and Asia, et cetera. Actually, they do. But at the moment, so they're actually different mutational trajectories in the US and Europe. So it looks like the virus, to the best of our understanding, the virus started in China, sometime in the late 2019. And the history of its early spread is difficult to resolve because there wasn't a whole lot of sampling, but you basically had sort of different lineages get into the Western United States and in Europe, and then from Europe into the Eastern United States. But then what happened in the United States is that the lineage that got into the Western United States mostly died out. And then you had a new lineage that arose in the United States that's separate from everything else. So at the moment, we have reasonably separate geographic distributions, primarily because, all the travel's been shut down. As far as I know, you still can't go anywhere without quarantine, and it's really, painful. So we're kind of sequestering the virus to where people can travel. So it's not, but the differences between, but they're very minor differences. I mean, we're talking about 4 or 5 mutations in the entire genome. So as far as, are those viruses different clinically? Probably not. They're just sufficiently different that we can tell that they had a different history getting to the United States. Would environmental factors in different areas lead to any of these mutations? Not at this stage, I don't think so. It's all just, spread, propagation, and randomness. You know what I mean? Like summer versus winter. Yeah, I don't think so. Oh yeah, so as far as seasonality, so again, I'll address this in more detail later when we sort of talk a little bit more about epidemiology, but at this stage, it's all great speculation, right? I mean, a lot of the predictions were made about, for example, declining spread in the summer, and it didn't happen, say in the Sun Belt in the Midwest in the United States. I think a lot of it is now confounded with, various non-medical interventions, so, lockdowns, and it's really difficult to disentangle. But we'll talk about that, later. Yeah, so I mean, you can tell whatever story you want, right? So people stay indoors more during the winter, so transmission might decrease. But on the opposite, on the flip side of the story, people don't get enough vitamin D because they don't see sunlight, right? And you can pull out evidence that says that if you need vitamin D, it's just general health and immune robustness. If there's one thing that's true about coronavirus is that it's very tempting to pick one factor and say it's the important factor, but it seems like there are a lot of confounded factors that all kind of feed into one another, and it's really difficult to, at this stage, say what is actually driving this transmission, and why we're seeing things that we're seeing, because we do know, for example, that a lot of the early epidemiological predictions were way off base, right? So they were too pessimistic, and that's a great thing. I mean, if you go back, we'll do this. I'll go back and show you predictions that people made in March, and then you'll be happy that these predictions didn't materialize. So we didn't have 5 million dead in the US as some of the models were predicting. So pre-existing immunity, what does it mean? So there's this fairly short but somewhat dense paper in Science that came out fairly recently. It's entitled Selective and Cross-Reactive SARS-CoV-2 T-cell epitopes in Unexposed Humans. So the laboratory component is very complicated, and I don't pretend to understand. I wouldn't be able to do it. I have an understanding of what they did, but these are great immunologists. I know these two guys I used to work with, Bjorn Peters and Shane Crotty. They're really good immunologists from UC San Diego, La Jolla Institute of Allergy and Immunology. So I worked with them on HIV. So they're good immunologists. Of course, I should trust but verify. But this is a good paper. So the goal of the study was to see if people that had never seen COVID or SARS-CoV-2 had any immune responses to the virus. And the hypothesis, going into the study, and that had some preliminary evidence from previous studies, is that exposure to common cold coronaviruses, that if you remember from 2 lectures ago, have similar proteins, could provide protection against SARS-CoV-2, right? So the idea is if, we were all infected with some form of coronavirus as a common cold, quite likely, especially, if you've lived for a couple of decades, and your immune system cleared that, nobody, or almost nobody, dies from complications from common cold. So your immune system was able to develop a response, and there's probably a memory immune cell that remembers something about it. So it's not exactly what you would see in SARS coronavirus, but it might be similar enough because those viruses are genetically related. Now, they wanted to do it on people that had never seen COVID SARS-CoV-2. So what they did is look at samples that were collected before COVID. So the stored samples from 2015 to 2018, and these are so-called PBMC, not PMBC, peripheral blood mononuclear cells. So that's a common, type of cell that's used for immunological studies. And then they performed experimental screening to determine if any of these cells recognize any of the SARS-CoV-2 epitopes. And they took a brute force approach, so just parallel screening. They split the SARS-CoV-2 parts of the SARS-CoV-2 genome into 474 15-mer peptides. So it was this is basically called tiling the gene. So you take it and you chop it up into pieces, overlapping or non-overlapping. There's some strategies of how to do this, but basically you just hypothesize that you sort of give the immune system a large number of potential targets that it might see during a SARS-CoV-2 infection. It's not all the targets that that SARS-CoV-2 present. They're just representative, and there's a large number of them. And then you basically screen them through a procedure where it's an in vitro procedure where you do it in cell lines, but you stimulate, not cell lines in patient cells, but you stimulate naive T cells for a couple of weeks, I think 2 weeks, and then you measure whether or not they bind this peptide. So it's a very well established complex laboratory protocol that is, takes a lot of time to do, especially if you want to do it in a lot of patients, a lot of different epitopes. But they completed it and identified 142 epitopes that were recognized. Out of these, so about a third of the ones that they tried were recognized. 66 were from spike. The rest were from other genomic regions. Interestingly, each of the donors recognized a median of 11.4 epitopes, so it wasn't focused on just one, it recognized several. Every single individual recognized at least one, so there wasn't a whole, there wasn't a single individual that recognized nothing. And interestingly enough, 40 epitopes were recognized by more than 2 donors, which means that there's some commonality to the immune response. So this is based on simulating T cells and creating a de novo response, right? So this this basically is kind of simulating what an infection looked like. The only twist is that you're doing it on people that could have not possibly seen COVID because, you know, they're historical samples. And, you know, they observe there's really no preference for particular peptides. So if you look at, as the protein becomes longer and longer, it has more peptides that are recognized. So it seems like the entire viral proteome is immunogenic. So all it tells you so far is that, you know, healthy individuals that have never been infected with SARS-CoV-2, if they were infected with SARS-CoV-2, they would all raise a detectable T cell response. So there's nothing about cross-protection here at all. It's just that you have the ability to respond to SARS-CoV-2 infection. So, but what they also did as a side note is that they observed, right, so this de novo development takes some time. So you need to stimulate cells and you need to wait for them to go through the appropriate you know, differentiation, maturation cycles. So it takes some time, it takes 2 weeks. But they also observed, you know, independently of this, there was some reactivity before any of this happened with memory, which is consistent with memory response. So that means that you don't have to develop the response de novo. It means that there might have been some memory T cells that recognized something similar from a previous infection that those people had. Then they attempted to ascertain that this was the case. And they observed that, so the first thing they observed that responses shared by more than one individual were to epitopes that were more similar to other coronaviruses. So here the logic goes a little bit like this. If you had multiple individuals target the same epitope in SARS-CoV-2, perhaps this is the same kind of epitope that would have targeted in a common cold infection. So here's, you know, these are, if you've never seen these plots, they're kind of ugly violin plots. So they're meant to show you, you know, distributions. So this is, and what they're trying to do here is they're trying to say, among the SARS-CoV-2 epitopes that were recognized, how similar were they to epitopes in other coronaviruses? So for example, you know, here's a band that says these epitopes were not recognized by anyone. So there are 332 epitopes that didn't actually raise any responses. And the median similarity between those and other coronaviruses was 40%, the amino acids. Then you go to one individual, doesn't look any statistically different. If you go to 2 individuals or more that target the same response, this is now about 50% similar to some coronaviruses. So inference here is that if an epitope of SARS-CoV-2 looks more like what's being seen in other coronaviruses, then more than one individual will raise that response. And here's another way to look at this, at you know, strong response versus weak response. So basically now they're saying, look, among the epitopes that recognize, knowing nothing else, they seem to look more like the epitopes that are seen in other coronaviruses. So now let's do a direct experiment to you know, conduct a direct test of cross-reactivity. So I'm already over time, so I'm going to try to go through this quickly just to give you the take-home message. So they synthesized even more peptides. So what they did, they took the epitopes that were recognized by SARS-CoV-2. Then they tried to identify sort of matched epitopes in other coronaviruses. They generated a new library of those. And then they found an additional set of individuals not involved in the original screening, you know, from the past to do this test on. And they also screened the response to other coronavirus epitopes in people that had COVID-19 and recovered. And these are busy plots, so let's see if I, we'll just go through one, and then you can sort of look at the others on your own. So this is looking at unexposed individuals. Each bar is a peptide pool, right? So here you have DMSO, which is a negative control. Here you have proteins, here you have peptides split by whether or not they're from spike or non-spike. And these are different libraries. So R129 and R30 are peptide libraries that are from coronaviruses that are not SARS-CoV-2. And CD4R is peptides from SARS-CoV-2. So what you see here is that if you take epitopes that were recognized from SARS-CoV-2 and find matched epitopes from other coronaviruses that are also recognized, they're more recognized in SARS-CoV-2, but they're also recognized from other peptide pools. And it's the same between unexposed and COVID individuals, except in COVID individuals, there's a more robust response because presumably they've more recently seen it. But here's what the, and finally they did direct tests of cross-reactivity. So they took specific cells, and this is a very, very involved laboratory technique because you actually have to cell sort. You have to identify cells that are specific to antigens, and you have to, put them in separate cultures, do multiple replicates. But here's basically what this shows to you is that, this is a particular epitope from, say, a particular individual. Like, so let's look at panel B. So this is some epitope S816 from donor 2209. Here are the epitopes that they screened. So this is what the immune system actually recognized, these particular lengths of amino acids. So SARS-CoV-2 is the SARS-CoV-2 peptide. And what's below it are peptides that are different, because they're from different coronaviruses, but they're still similar. So for example, this OC43 peptide is 73% identical to the SARS-CoV-2 peptide. So they screened all of them. And what you see here is sort of a dose response curve. So how much do T cells respond to progressively decreasing concentrations of these epitopes? So here's sort of the reference concentration 1, and the higher this is, the more T cells are activated and respond to this particular peptide. So you can see that everything except OC43, the 3 other peptides actually are also being recognized by the cell that originally developed response to this peptide. So this is a direct confirmation that if you saw this particular peptide, you would also have immunity against other coronaviruses. And they did a number of confirmation studies to see that this is true. They also, again, I'm going to skip this, but basically they also confirmed that these were the correct type of T cells, so memory T cells. I might come back and do it in the beginning of the next lecture again. So to summarize, SARS-CoV-2 contains a large number, 140 here, epitopes targeted by T cell response. They also showed that SARS-CoV-2 reactive cells also cross-react with epitopes from similar coronaviruses. This is sort of the meat of the paper, where the same immune cells that are responsive to SARS-CoV-2 are also responsive to epitopes from other coronaviruses. And that is a behavior, this is called cross-reactivity, when the original response was to one virus, but then an infection with a different but related virus still triggers the immune response. And this is different from antibodies that appear to be highly specific to SARS-CoV-2. Cross-reactivity to CTL epitopes could contribute to modulating disease severity, right? So, and this is, again, an important point that I would like to stress, is that T cell responses do not stop the infection. It means if T cell responses are active, it means you already have the virus infecting cells and replicating. What they do, they can kill these infected cells. And if you don't start from 0, if you start somewhere, from memory T cells, you can do it much faster, which means that the disease that could have been severe is now asymptomatic or mild. All right, let me